Good evening, students. So let's start our today's session. So welcome everyone to the NPTEL course on novel technologies for food processing and shelf life extension. And This course is instructed by Professor H.N. Mishra, Department of Agriculture and Food Engineering, IIT Kharagpur. I am Shikha Sharma, a research scholar in the Department of Chemical Engineering, IIT Guwahati. I'll be the TA for this course for 2025, January 2025 semester. And this is a live interaction session in which this is a week 2 session, and it is on the topic chemical changes in food during processing, browning reactions, enzymatic and non-enzymatic, protein interactions, carbohydrate interactions. I hope you have already gone through the 5 lecture videos of the week 2. And in this live interaction session, we will be practicing some of the problems and concepts behind to the week 2 concepts. And we will trying to solve some of the questions, the practice problems today for the week 2 topics. And if you have any doubt regarding the lecture videos and the live interaction session, please feel free to ask. So also if there is any internet issue or if there any sound issue, please let me know at that time so that it do not interrupt the further session. So yeah, today's topic of the discussion are browning reactions, food proteins, principles of the food preservation, traditional food process preservation technologies. So let's begin with our first first question, which is the like the conceptual basis question. So which one of the following generally does not participate in the enzymatic browning reaction? So option A is diphenol oxidase and tyrosinase. Option B is yes. So option A is diphenol oxidase and tyrosinase. Option B is phenol oxidase and phenolase. Option C is acetaldehyde dehydrogenase and succinate dehydrogenase. Option fourth is monophenol oxidase and oxygen oxidoreductase. So this is our first question for today. Anyone would like to, because you have already gone through the lecture videos, if anyone would like to attempt. Hello, anyone? Am I audible or not? At least let me know.
Yes, ma'am.
Yeah,
You are
ma'am.
audible.
Yeah.
Okay, okay. So let's try to solve it. So the option C is correct, which is acetaldehyde dehydrogenase and succinate dehydrogenase, because it does not participate. Yeah. So it does not participate in the enzymatic browning reactions. What happens? These two enzymes, the acetaldehyde these two enzymes, the acetaldehyde dehydrogenase and succinate dehydrogenase, they basically uh, they are they do not participate in the enzymatic browning because this enzyme, the first one, the acetaldehyde dehydrogenase, it basically its function is to it converts the acetaldehyde into the acetate. The like in our body, when we drink anything like which has ethanol or alcohol, then what happens? The alcohol dehydrogenase enzyme converts the alcohol into the, like in this example we can see in the figure on the left side, we can see that the ethanol, when we have the ethanol, the alcohol dehydrogenase enzyme, it converts it into the acetaldehyde, which is a toxic product in our body. So what happens in the mitochondria? The ALDH, which is acetaldehyde dehydrogenase enzyme, is present, which converts this acetaldehyde into the first the like the acetic acid, and then further this will be converted into the CO2 and the water, which are the non-toxic compounds. So this, since it is its function is something else, so it is a like non, it doesn't participate in the enzymatic browning reactions. Second is the succinate dehydrogenase. So what it does do is it oxidizes succinate to the fumarate, and then the fumarate will be further reduces the quinones as a compounds of the TCA cycle. That in the TCA cycle, this enzyme, the succinate dehydrogenase, it oxidizes the succinate to the fumarate and then reduces to the quinones. Like we can see in the TCA cycle, here this enzyme is there and it is reducing the succinate into the fumarate. And then in the electron transport chain, its function is to reduce ubiquinones to ubiquinol. So since it is reducing here, the second function, it participates in the electron transport chain. So it is not participating in the enzymatic browning reactions. So our option C is correct here. Other enzymes, they participate in the enzymatic browning reaction, like the enzyme polyphenol oxidase. We hear its name very popularly. This enzyme is also known as the phenol oxidase and the phenolase, also monophenol oxidase, diphenol oxidase, triosinase, right? This is its name, other names in other forms, and it also participates, like it has also functions as a enzymatic in the enzymatic browning reactions. Also, the phenol oxidase enzyme, it is responsible, it catalyzes the oxidation of the phenolic compounds to the quinones. So the phenolic compounds, it will oxidize and convert into the quinones, and then which further converts into the melanin, which is a colored compound. So the image is not very clear, but I'll tell you that what happens. Tyrosine, it converted into the dopa. Here, the enzyme which is phenol oxidase, which is causing the enzymatic browning reaction, which participates in the enzymatic browning reaction. So the tyrosine compound in our food, it converts into the dopa, and then which is further catalyzed by this enzyme phenol oxidase and convert the dopa quinone, which is further converted into the leuco compounds. After that, these compounds are converted as dopachrome, and further different reactions will be there, like quinones will form, indoles will form, and at the end melanin will form, which is having the colored property, brown color it imparts. Sometimes it forms in the food as in product, like we say that enzymatic browning happens because mostly this kind of somewhat this kind of reaction happens in the food. And polyphenol oxidase, it is the main enzyme which do enzymatic browning. So all these other options, they cannot be, like, they participate in the enzymatic browning reaction, so they are, they cannot be correct, right? Is it clear?
Yes, ma'am.
Okay, thank you. So second question is, match the reactions. Here in the column 1, there are several reactions uh, or the processes, like darkening of apple surface on peeling. Second is the Maillard reaction. Third is the loss of flavor. And here in the second, its type is given, like, is it a physical process or a chemical or enzymatic process? So let's try to understand what is happening. So option C is correct, which says that one is related to R, which is the darkening of the apple surface. It is related to the enzymatic process or the enzymatic changes. Maillard reaction, it is related to the chemical type, and loss of flavor, the physical one. So what happens? There are different processes which happens, like uh, which can be characterized as, like, it can be due to microbial changes, enzymatic changes, chemical changes, or the physical, because of the physical changes. So like in the microbial changes, what happens is uh, there will be growth of any toxic substances, and like the microorganism is producing that, either like infectious microorganism or the spoilage causing microorganism. They basically grow, and because of their presence, food may get infected. In the enzymatic side, it's hydrolytic reactions, which are like uh, catalyzed by the lipase or the protease enzymes. So these enzymes, they catalyze some of the reactions which cause some of the uh, reactions in the food, like we just now discussed about polyphenol oxidase. This enzyme cause the browning, like lipooxygenase enzyme, the enzymatic brownings, all are because of the enzymes are participating. Because of that, it is happening. And third is the chemical processes, which are like oxidative rancidity, oxidative and reductive discoloration, non-enzymic browning, and the nutrition loss. So all these happens because of the chemical changes happen, or chemical reaction takes place. Fourth is the physical changes, like because of the, like, mass transfer, like the moisture is transferring from the food to the atmosphere, so there's a moisture loss, and because of that, sometimes the texture affects, the movement of low molecular weight components, like transferring of the volatile compounds from the food, which results in the loss of flavor. So, and as a loss of crispiness or the texture, which may be because of the water loss, or maybe because there is the enzyme, like because of the enzymatic changes also, like pectin methylesterase, it degrades the cell wall of the fruit, and which leads to the decrease in the firmness of the fruit. And in the physical changes, like freeze induced damage may happen, like when we like it's the when we do, there is a temperature abuse, like when there is a low temperature and suddenly we shift um, to a higher temperature, and so much of the temperature fluctuations will be there. Then uh, because of the sudden change in the temperature, chilling injury, or when we do thawing, there is a loss of the texture of the food product. So it is because of the physical changes. So the darkening of apple, it's because of enzymes, because enzymes act browning, enzymatic browning. And Maillard reaction is basically a chemical kind of process, and the loss of flavor or the loss of texture, it can relate to the physical type. Is it clear to everyone?
Yes, ma'am.
Yes, ma'am. Yeah.
Okay, thank you. So we are moving to next question, which is during non-enzymatic browning, the reactive reducing sugars, it undergoes upon heating. So opening of the ring structure, then dehydration, dehydration followed by enolization, enolization then fragmentation, and fourth is a ring opening, enolization, dehydration, and the fragmentation. So it is being discussed in the Browning reactions lecture slides, lecture videos by the Professor Mishra already. So the option D is correct, which is ring opening, enolization, dehydration, and fragmentation. So what happens? The reactive reducing sugars, they are basically countable for the distinct color and the flavor in the foods, like in the dates, honey, chocolate, etc. So it results in the non-enzymatic, like all these uh, happens like their distinct color and the flavor is coming because of the non-enzymatic browning. So these sugars, because when they are heated, so it the sugar will going to be in the process of its ring will going to open, and then enolization, and then enolization, like what happens in the enolization, the carbonyl compounds, basically carbonyl compounds, like esters, ketones, and aldehydes. They are basically they are converted into the enols form. So that is known as the enolization. This carbonyl compounds, they basically convert into the enols. And then dehydration and fragmentation. So the sugar, it can go like when we heat sugar, it can go any in the any of the process, like ring opening, enolization, dehydration, or the fragmentation. And because of that, there may be generation of some of the distinct colors, like the caramelization of is one of the examples. So distinct color or the flavor, like in the maybe in the dates, honey, and the chocolate, we can see there is generation of different kind of a unique color or the flavor, which uh, mm, which adds to the uniqueness of that product. So it is because of the non-enzymatic browning reactions. So option D is correct here. Is it okay?
Yes, ma'am.
Okay, ma'am.
Okay, thank you.
Yes, ma'am.
So next is which of the following is not a part of the non-enzymatic browning reaction? So out of all these, which is not a part of the enzymatic, non-enzymatic browning reaction? Uh, so let's see. Beckman rearrangement, it's not a part of the non-enzymatic browning reaction, because what happens? It's a chemical process where an oxime group is transformed into into substituted amides group, a technique utilized in the manufacturing of the nylon. So when we make the nylon, in that there is a chemical process where an oxime group, it is transformed into the, like it substitute with the instead of that one amide group will come and attach, which forms the nylon. And in Stacker's degradation, what happens is the oxidative breakdown of amino acids will happen by the carbonyl compounds. So because of that, there is a degradation form of the ketose amines will form, and the amino acids, they will react to form the, like they will react and they will form the skip bases first, and then they will go in the acid catalysis reaction, decarboxylation reaction. So this is the Strucker's reaction, Strucker's degradation. In Strucker's degradation, it happens like this. So third is the, like when our ketose, like the fructose, which is the sugar, is reacting with the amines, amino aldose, they are formed, and this reaction is known as the Heinz reaction. And fourth is the Amadori rearrangement, where an aldose, they will basically form the ketose amine. So when the aldose, which is a, like is glucose, and the amino acid is glycine, the amidary product like this MFG, which is monofructose glycine will going to form, like one amino, one deoxy, two fructose. So all these reactions, the Strecker's degradation, they are basically they are basically involving some kind of the they are going to form like the no, they are non-enzymatic browning they are causing. There is like no use of any enzymes in their reaction. In amidary reaction also, this is purely a chemical reaction, right? And the Heinz reaction is, it is also. But in the Bachmann rearrangement, in this is also like this is a chemical reaction, but it doesn't it doesn't produces the browning reaction, like browning substance. It is used for the nylon production. So option B is correct here. So is it clear?
Yes, ma'am.
Yes.
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
Yes, ma'am.
So we are moving to the next slide. Next question, which is about again the non-enzymatic browning. So which of the following generated during non-enzymatic browning, which is 3-hydroxy 2-methyl pyrene 4 1, and second is 5-hydroxy methyl 2 foraldehyde 3, in third is 3-hydroxy 2-acetyl furan, and fourth is 2H4 hydroxy 5-methyl furan 1 furan 3 on. So from this, which option can be correct? So option 1, 2, 3, 4, which these all are option correct. Why? Because in the non-enzymatic browning, what happens? The sugar, the reducing sugar, it undergoes a reversible reaction where what happens? The amine group, it basically form the glucosyl amine. glycosylamine it will form, and which form the furan derivatives, where like we see like from the hexose there is the formation of the HMF, which is a furan derivative. And in the pyrolytic reactions of the sugars, what happen is unsaturated ring structure that contribute to the unique flavor and the fragrance it will like form. Also, they will impart some of the color. Such compounds are like maltol and the isomaltol, and their chemical name is given here. So in the options, all the their like common names are not given. The chemical names are given, and this maltol, isomaltol, they are adding the flavor in the bread. And the another compound, which is the our last option, if we see this last option, hydroxymethylfuranol is utilized, and it is giving the like enhance the flavor or the sweetness. So in the reaction, if we see what happens, the reducing sugar like the glucose and the amino acids, they undergo to the reaction and form some like compounds. It goes to the amidary rearrangement and others like that in and finally it forms some of the compounds like which give the color, the distinct color or the flavor to our food product. Like in the bread there is maltol and isomaltol. So these compounds all are generated during enzymatic browning, like this, like these two, which is option number uh, this third option, and the first option, it is generated in the bread. The first option, which is the
Ma'am, I think you're
sorry,
not audible. You got stuck, I guess.
hello, am I not audible?
Audible, madam.
Audible, ma'am.
So I think maybe you should check your sound. Yeah. So all these are uh, in there, like all these compounds, they are present. Yeah, all these compounds, so they are somewhere or other they are involved in the formation, like in the process of the non-enzymatic browning, and they are contributing to the colors or the flavor. So all the options are correct. Is it clear?
Yes,
Clear.
ma'am.
Yes,
Yes,
ma'am.
ma'am.
It's like very much chemical reactions in the browning, the browning reactions lecture. But yeah, it's like important reactions. So in the food, so we need to understand. So next is the when we do the controlled Maillard browning reaction, then which in like which kind of product we get, and the desirable product we get. I think this one is easier.
Ma'am, bread.
Okay, so we have one person who is who said bread.
Auntie Bali.
Okay. I'm not able to hear you clearly.
Auntie Medina.
Kanthimati, yeah. So one is saying bread. Anyone else? I'm showing the slide again. It's easier, right?
Canned food.
Canned food. Okay. We have two options.
Bread, ma'am.
So
Canned food.
okay, we have more votes towards canned food.
Bread. A.
Ma'am,
Bread.
bread.
Bread. Okay.
Bread.
So many are uh, telling about. Is it possible to extend the assignment submission date of week 2? I'm not like it's sorry, but the assignment submission dates are fixed, and you need to submit on you need to submit on time. Assignment 1 answer, please share. So it will be like under checking. It is under checking, and uh, it is not like handled by me, so I cannot share the assignment 1 answers. You can ask on the discussion forum such questions like regarding the examination and regarding the PDF of the lecture videos. Such questions you like I also asked, and they told me that instructed me that please ask students to ask such questions on the discussion forum so that everyone can get benefit out of that. Is it possible to? No, no, not possible. Okay. So we are moving to our question. So the answer is A, bread. What happens? The Maillard reaction happens in all of these, but because in the bread there is development of the brown crust. There is development of the brown crust during the baking, which provides its characteristics color to the bread. So, which is a desirable change happens while the brown color happens in the the juice, orange juice, or the dried fruits and vegetable and the canned food. The reaction happens, but what happens? It's not desirable. It comes under the reduction in the quality. So bread is the correct option here. So its highlight is desirable here. So we are moving to the next lecture video about the protein. So we are moving to those questions about the protein. First is which option accurately describes the quaternary structure of the protein? A sequence of amino acids in linear arrangement, consistent structural motifs like the alpha helix and the beta sheets. Option C is comprehensive 3D structure where there is interplay among various subunits. Fourth is three-dimensional configuration of atoms within the polypeptide chains. The quaternary structure, it's very simple, right?
Ma'am, C.
C,
D. D.
B,
Ma'am, D.
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 D, D. Okay,
D.
B, okay,
D.
B. Someone is
C.
also
C, ma'am.
saying B. Let's see the answer. C is the correct answer. So what happens in the primary structure, which is the simplest structure of a protein, it has a linear sequence of the amino acids, which is connected by the peptide linkage, right? Here, this is amino acid, and these amino acids are connected by the peptide bond, and this kind of linear sequence we get, right? Which is the simplest and the primary structure of the protein. And in the secondary structure, what happens? There are like regular some kind of arrangement is there like it is the alpha helix and this is the beta sheet, the bottom one. So in there, what happens because of the formation of the hydrogen bond, it like between the NH here, like we can see NH and C double bond O of two different amino acids, they are forming the hydrogen bond, and because of that, there is the helical like structure. And in the beta sheets, what happened? Uh, it arranged in the form like sheet, so where it can be C to N terminus or N to C terminus. So in the tertiary structure, what happens? There is three-dimensional arrangement of atoms in the polypeptide chain. So here it's the polypeptide chain. So there can be ionic bonds, hydrophobic interactions, disulfide linkages, hydrogen bonds, and because of that, the three-dimensional arrangement, because of these bonds, such arrangement like it forms such arrangement. And in the quaternary structure, what happens? This kind of like tertiary structure will be there, and there are so many like that, and there will be like 3D interaction between the protein subunits. These are the subunit, one subunit. This is the one subunit. So there will be many subunits like this. So option C is correct. In the uh, three-dimensional interplay among various subunits. Is it okay?
Yes, ma'am.
So we are moving to the next question. In the column 1, there are different properties like textural properties, structural, rheological, hydration. In the second, there are different functional criteria like gelling, elasticity, network, cross binding, and viscosity. So the textural is because of like which functional criteria like this. So option B is correct, which is saying that the textural is connected to the network cross binding, and the structural is because of the elasticity, like it is the property because of the elasticity, like the structural related property is elasticity, and 3 is rheological property, which is related to the like viscosity, and the hydration is about like the gelling. So what happens? There are different properties, and there are different their functional criteria. Like we can see the surface property. What are the surface property? Like because of the surface, there will be either emulsification or the forming, or there is a kind of film formation. Like this happens because of the like at the surface of any like surface of the molecules, like forming. In the forming, what happens actually? There is air to liquid interaction, like at the surface tension will gonna decrease, and the air will be trapped inside the liquid, right? So, and this is a structural, right? Its elasticity, I doubt it's emulsification. It should be. Then textural properties. Textural properties is about the adhesion, the network cross binding, aggregation, gelation, dough formation, texturability, fiber formation, extrudability. So all these are related to the texture of the product. Surface and the structural, these are two different things. So here it's structural. I'm sorry that I said surface. I misunderstood as surface. It's structural. So structure is related to the elasticity, grittiness, cohesion, and chewiness. So it's because of the structure. If there like it can be either like elastic. Gritty means like it's like sand. Cohesive means like forming the with the same molecules, it how it forms the bond. And chewiness is how it is going to chew. All these are the structure, how depends on the structural property. And the rheology means how a fluid is flowing, right? So it depends on the viscosity or the gelation. Enzymatic means like different enzymes we have, like the rennet, which coagulates the milk, and the papain, which tenderizes the muscle. So all these are related to the enzymes. Surface, at the surface we have the emulsification also, and the forming. So in the emulsification, what happens? Um, there will be formation of like two emulsions, which is oil in water and water in oil. And binding, in the binding, lipid binding and flavors, they come. And in the hydration, there is like different properties, like how it behaves in the water, like solubility, wettability, water adsorption, swelling, thickening, gelling, viscosity. And in the kinesthetic, it can be about the mouthfeel, texture, smoothness, grittiness, and the turbidity. So all these are the different functional properties which is related to the functional. It's like these are the properties which has their functional criterias like this. So option B is correct. Is it okay?
Yes, ma'am.
Okay, ma'am.
Yes, ma'am.
Okay. Okay. So next question is about, among the given options, which has the least or negligible impact on protein denaturation. So all these affects the protein denaturation. Which one is the least? The heat, charge, pH, or organic solvents? Anyone would like to try?
Charge.
Organic solvent.
Charge.
B.
Which one? B. pH. The least negligible impact.
D. Option D.
D. Organic solvents.
Organic solvents, ma'am.
Organic solvents, ma'am.
Okay, so many are saying organic solvents,
Option D,
right?
ma'am.
Option D. So let's see. Option B, charge. Because protein denaturation is affected by the temperature, pH, and the in which solvent it is there mostly, and least by the charge, the minimal effect because of the ionic charges. Okay, because protein, how if it's there in any kind of solvent, then it's the most
Ma'am,
effect. Like yes,
ma'am, if question ask most impact on protein,
all
then
like the
answer
heat
H.
and the
Heat is the
pH.
heat.
Heat is also there, pH is also there. If even if you uh, do slightly change in the pH, it the protein will going to denaturate and it unfolds, right? Even in the solvent, it has a good impact, because the solvent, if in the solvent we say that like we some are water soluble, some are like uh, organic solvent solution, so like the soluble. So if it's not as per the uh, structure of the protein, how it will going to solubilize, not right. So all are actually important, not we cannot say that most one, like
Okay, ma'am.
yes, uh yeah. So heat is also like heat and pH actually mostly we see the effect of heat and pH, and even if there will be slightly change in the pH, it like the protein become insoluble in even the solvent which it was before soluble. So we cannot say one as most, but all are important factors because protein is a very complex and sensitive kind, like you can say. Okay, so is it okay? Am I able to answer?
Yes.
Okay, okay. So next is the which among the given options represent a structural characteristic of the protein? Emulsification, coagulation, cohesion, and gelation. So
Ma'am,
structural
option C.
characteristic, cohesion.
Yes, ma'am.
Yes. So cohesion is
Ma'am,
the correct
coagulation.
answer.
Ma'am,
Coagulation knows. It's
Coagulation
coly.
should be, ma'am, because when
Mm,
the egg albumin is heated, it coagulates.
but it's like mostly coagulation is related to the enzymatic property, like uh, but not the structural property. And cohesion is basically because the all the cohesion, how two molecules they are like interacting with which force it is interacting. It all depends on the like cohesion, right? Even like we say that two molecules, if it is having like how much apart they are and with which force they are, with which size they are, all these they come into the account of the cohesion, right? So the cohesion, elasticity, grittiness, and chewiness, they all come in the structural properties. While emulsification, it comes in the surface property, and gelation, it comes in the rheological property, and coagulation is the enzymatic property because it coagulate because of rennet is an example, like in the milk in the proteins, milk proteins, they coagulate because of the presence of the rennet enzyme, right? Is it okay?
yes, ma'am.
Okay, ma'am.
Yes, ma'am.
Ma'am,
Yes.
ma'am, today I submitted assignment 1, week 1.
Mm-hmm.
Marks are not uploaded.
Yeah, but it will be uploaded all together, right? It's the deadline is 5th, right?
Ma'am, please repeat again. Network error.
I am saying all will be uploaded together.
Ma'am, I have one question, ma'am.
Yes.
Ma'am, how to submit assignment, ma'am? Is it in online?
Yes, online, right? The submission button is there.
Oh,
Mm-hmm.
okay, ma'am.
Madam, this is objective type or descriptive type?
In the assignment, the questions are there to solve, and like these options are there. Like these options are there. You need to understand why you are correcting, yeah, why you are choosing that particular option. So you should know the concept behind, right?
Right,
So
madam.
it's like you are selecting, but you should solve at your notebook, right?
Right, madam. Right.
So we are moving to yes, 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 please.
So, ma'am, it means
Yes, ma'am.
exam is objective type.
Please ask this question on discussion forum because I emailed and they told me that exam related question like you need scientific calculator, not how to submit an assignment or um, the like about the deadlines of the assignment or the like the PDF of version of the video lectures. You should please ask, like you should ask on the discussion forum with a bold heading like such particular for that particular query. I think that, and because if they answered there, it means if we are answering there, it means everyone is like going to understand that what is happening, right? Please write your query on discussion forum, such queries.
Okay.
Hmm.
Okay, ma'am.
Okay, so we are moving to our question. The time needed for half of the liquid to drain out of a foam or for a 50% decrease in the foam's volume is utilized as a measure to express foam stability, foaming capacity, foam reduction, foaming power. So its answer is foam stability. Why? Because the foam stability, its answer I have also like explained one person's query on the discussion forum about the formability, about the forming. It's a detailed answer. So please everyone read that answer also. I'm here, I'm describing, but you should also read that answer. I will suggest. So foam stability, it basically denotes like the foaming is basically denoted by the capacity means which how much foam or how much air it can entrap, and stability means the capacity of the foam to resist the gravitational and mechanical forces, which means that if it resists those forces and it can be like the foam will be there, and it like after if we see at time zero such this much height of the foam forms, right? After some times there will be some reduction in the foam and the height will reduce because of the gravitational and the mechanical forces. So the forming stability is the time which it takes for the half of the liquid to be drained from the foam. Like we see the time, we calculate the time in which the height will going to be half, or there is a foam volume which is reduced by 50%. So the option A is correct here. Is it okay?
Ma'am, the reason for the
Yes, ma'am.
reduction of foam volume?
Because of the like what happens when the foam is forming, right? So there will be some kind of gravitational forces which will try to if something is on height, then there will be gravitational forces because of the weak forms. It will reduce its height, and also because of the shear forces among the uh, air bubbles, it decreases, right? So after some time its height will decrease.
Okay, ma'am, got it. Thank you.
Hello.
Yes, ma'am.
Yeah, so is it clear? Also, I've written that detailed answer. Sometime what happens, the smaller bubble, it also because this because if the smaller the bubble is, the higher the internal pressure will be there. So what happens? Sometime it drains, and the it collapses like that. It the like it like that because it has the internal pressure so high that it tries to break to release the pressure.
Okay, ma'am, now I got it.
Okay,
Thank you.
thank you. So we are moving to the next question, which is among the listed options, which are the extrinsic factors that influence proliferation of microorganisms in food. So first option is freeze concentration and oxidation potential. Second is oxidation and reduction potential. Third is equilibrium relative humidity. Fourth is and reduction potential. Fourth is equilibrium, relative humidity, and temperature during storage. Extrinsic factor.
Ma'am,
Option D.
option D.
D, ma'am.
D.
Yes, ma'am.
Okay.
Yeah, ma'am.
Yes, D, ma'am.
Okay. So let's see. Yes, you people are correct. So the growth of the microorganism is basically affected by two factors, which are the extrinsic factors and the intrinsic factors. So the equilibrium, relative humidity, and the temperature during the storage, they are the extrinsic factors, and the reduction potential and the freeze concentration, the oxidation reduction potential, they are all the intrinsic factor of a food. So option D is correct. Next is determine the microbial survival curves labeled as 1, 2, 3, 4, 3 in the given illustration depicting three different types of microbial survival patterns, which is like thermosensitive and thermoresistance microorganism, coexist, homogeneous heating, and germination of the spores. So these three are in option A. So option B is saying first is homogeneous population, second is microorganism activated by short exposure to heat, and third is saying thermosensitive and thermoresistant microorganism coexist. Option C is first graph, this one. This is depicting composite population of cells. Second is microorganisms deactivated by the short exposure to heat, this one. And third, germination of spores. Fourth is saying first is homogeneous population, second is thermosensitive and thermoresistant microorganism coexist, and third is germination of spores. So
Ma-
anyone?
Ma'am, D.
D. Okay, D.
D,
Yeah,
ma'am.
ma'am. Yeah.
Okay, let's see. D is correct. Why you said D? Anyone would like to explain? Your answer is correct, but can you like try to explain the reason? I will also add in that. Anyone? Okay.
Because ma'am,
Hmm.
ma'am क्वेश्चन पे एक बार जाना क्वेश्चन पे.
Yes. Yes.
Ma'am, here we are talking about the homogeneous population of the
Hmm.
microorganism. Means
Hmm.
have microbial activity, enzymatic activity, temperature, and all are
Hmm.
the factors are same for all
Hmm.
microorganisms.
Hmm.
So we can coexist.
In fun. Hmm.
Yes, ma'am.
Hmm. And in
According to me,
thermosensitive,
this is the
and in like in the second curve, why you are saying like it's thermosensitive or thermoresistant microorganism coexist, and in third like germination of the spores. So it's like the relation between the slow survivors, microorganism survivors with respect to the time. So it's a
Hmm.
semi-log curve, right? In
Yes,
what
ma'am.
happens, yes. So in what happens, uh, we are seeing that how many survivors means how many microorganisms they are surviving when we are applying the heat and with respect to time. So if there is the uniform population of the microorganism, then uh, it will be constantly like decreasing, whereas we increase the time with like constant temperature. If we increase the time, so it will with the passage of the time, the microorganism, they will continuously going to decrease the population. But if it's a mixed kind of population, which are like the thermoresistance and the heat sensitive, so heat sensitive
तो
will
तो अरे वे देख रहे तुझे कुछ दे दे खाने को, ग्रेप्स वगैरह। और मेरी क्लास हो रही है 6 बजे तक, लड्डू और भूखे रहेंगे।
please can you mute? I'm sorry, but like please mute because there was some other kind of discussion going on. Okay, so it's okay, sometime it happens. So in the heat sensitive microorganism, what happens, their population will suddenly gonna decrease, because they'll be in the contact with the heat, so their population will decrease. But the thermal resistant, which is heat sensitive, their population will not gonna decrease so like fast. It will decrease, but with a very low rate, because it can uh, it can survive that temperature. In the spores, which we know as that the microbial spores, they are basically they when they get the heat or they get any so what they are activated when we do the brief heat exposure. Like there can be the germination of the spores or the fragmentation of the chains. So it will be little like this. Usually our the full grown cells, they decrease like this exponentially with the time, and then they'll become constant, right? Yeah, this is not the semi-log curve, this is the normal curve, so it is like this. But if it's a semi-log curve, it will show like the one, how it's showing in one. Is it clear?
Yes, ma'am.
Okay,
Yes.
so we are moving to next question. Which among the below given methods is generally not utilized for decontamination in the food industry? Irradiation, H2O2, pulse light, pulse electric field.
H2O2.
H2O2 is not utilized for the decontamination in the food industry. One option. Next, anyone?
Option A.
Irradiation. Okay.
Irradiation.
Irradiation. Okay.
Option C.
Option C, pulse light. Anyone else?
Ma'am, A, C, and D are the actually the methods of used for uh,
Used for?
decontamination.
Yes, you are correct. So, he's correct. All these methods, irradiation, H2O2, pulse light, they are used for the decontamination in the food industry, whereas the pulse electric field, it is generally not used. So let's see why. Irradiation, like we do the irradiation for the like decontamination of the packaging materials. H2O2 also, the hydrogen peroxide is used in the aseptic packaging of the milk sometimes, and the pulse light also is generally used, but in the pulse electric field technology. It is in the like research. We can find many research articles on this, but if we see its application in the food industry, it's not generally not there because it has the limitation. And what is its limitation? Because there will be unavoidable occurrences of the electrochemical reactions at electrode and food interface of a pulse electric field treatment chamber when typical conditions for pulse electric field processing are applied. So when there can be some kind of condition because of when there is electrode and the food, they are like at the interface, it can form. Also, like these reactions, especially these lead to the corrosion, fouling of the electrode. They can also going to produce some kind of unavoidable occurrences, which is not good. And also the electrolysis of water will going to happen, migration of the electrode materials component to the food, chemical changes of the food product can also happen. So when it should be first minimized, and then the pulse electric food can be used as a decontamination in the food industry. Anyone is having doubt? Irradiation, I've seen like such, it is sometimes used like gamma radiation for the in the potato when there is a formation of the germination, or the sprout formation is there green color, so they are used to avoid this gamma radiations.
Ma'am,
Yes.
is radiation and radiation both are same term? We can
Irradiation,
use.
yeah, like I think it's same. Yeah.
Okay, ma'am. I confuse in this term. That's why I
Okay.
think.
Okay. I if any change to my statement will be there, I will tell you in the next class, but I think it's same.
Okay,
Most
ma'am.
anyone would like to contribute in this radiation irradiation topic? Please, it's a discussion, so anyone can put their opinion.
Ma'am, one question also.
Yes.
Ma'am, generally we can say
Mm.
decontamination in food cause
Mm.
change the chemical activity of a food.
Cause, can you repeat?
Ma'am, due to contamination of food,
Mm.
so that's cause chemical activity of food change. Is it
Contamination will
correct?
going to change, yes.
Okay, ma'am.
Ma'am,
Like,
to
yes, please.
so ma'am, this is what is coming from my previous work experience when we use the term radiation and irradiation. So
Mm.
referring to the malt storage for breweries, when a
Mm-hmm.
source emits a ray, let's say X7 X-rays emitted from a source, maybe a bulb,
Mm-hmm.
then that
Mm-hmm.
emission is called radiation. But if you
Mm.
expose something to that, then this process is called irradiation. So I think that's the interrelated term, and
Mm-hmm. I also think the same way, like it's the
the
same.
same.
Thank you for adding your views. So we are moving to the next slide. Which one of the following does not constitute an underlying factor influencing the microbial ecology in the food? Specific growth rate, antagonism, commensalism, and redox potential.
Ma'am, option D.
Option D we have.
Option D, ma'am. Redox
Option
potential.
D. Okay. Microbial ecology in the food. Can anyone going to explain like why the redox potential, why not like others?
Ma'am,
Mm.
option A, specific growth rate.
Okay.
It means if we provide the nutrient media for the
Mm.
microbial growth, specific
Mm-hmm.
growth rate occurs. So option A, eliminate. And option C is commensalism. It means two microbes maybe can be mutual, or one microbe have a positive relation and other have negative relation. So it does not constitute influencing the microbial ecology.
Mm.
And option C, antagonism. Actually, ma'am, this term is not, I cannot define it, but
Mm.
option D is we can eliminate because above three statements are similar, but redox potential is like as a
Mm-hmm.
physical term. So,
Okay. Thank you for your good answer. Redox potential is the correct answer. So, what happens in the antagonism? The term refers to an interaction between the organism where the organism benefits at the expense of another. But in the commensalism, it's a symbiotic kind of relationship where two organisms they were like benefit with each other. They'll not harm, but they'll help. Not the harm nor help. This kind of symbiotic relation. So these factors like the specific growth rate, mutualism, antagonism, commensalism are the implicit factor which are related to the food and that influence the microbial ecology in the food. But the redox potential, it is considered an inherent factor. It involves because of the like it involves the oxidation reduction kind of the reaction in the food. So where there is a electron transfer happens between the atoms or the molecules. So since it is an inherent factor and it is related to the exchange of the electrons, so it is not influencing the microbial ecology. But the others, it is related more to the like it is affecting the microbial ecology or the microorganisms which are present in the food. Is it okay?
Yes,
Yes,
ma'am.
ma'am.
Okay, so next question, it's the solving question. So if you like, you please try to solve with me. In a food sample, the starting concentration of the Clostridium botulinum was around 10 to the power 8 CFU per ml. Following a 1.5-minute heat treatment at 121 degrees Celsius, this concentration it decreased to 1 CFU per ml. So initial concentration was 10 to the power 8 CFU per ml and the final one is 1 CFU per ml. And we are applying 121 degrees Celsius for 1.5 minutes. We need to calculate the D value at 121 degrees Celsius. So we know that microbial survival curve looks like this when it's not in the semi-log form. So what happens when we do the heat treatment with respect to the time? So microbial population, it decreases with a very fast rate and then it becomes almost like constant uh, and it decreases continuously, right? So it is given in the question that the initial concentration is 10 to the power 8 CFU per ml and the final concentration is 1 CFU per ml. Treatment time is given and treatment temperature is also given. So We know that microbial survival curve is a first order reaction. It follows first order equation. So first order equation is the x equals to x naught e to the power minus kt, where x is the final concentration, x naught is the initial concentration, e to the power minus kt. K is rate constant or the death rate constant, because here we are taking that microorganisms are dying because of the exposure of the time temperature combination. So with the time microorganisms are dying, so we are taking its, calling its death rate constant. T is the time. So x equals to x naught e to the power minus kt. In this formula, we can put x is 1 CFU per ml, x naught is 10 to the power 8 CFU per ml, e to the power minus k into t is given 1.5 minute for like at 121 degree Celsius. So when we calculate, we'll get k equals to 12.28 minute inverse, like 1 upon 10 to the power 8 equals to e to the power minus k into 1.5. So when we do the log and do the calculation, we are going to get k as 12.28 minute inverse. In this question, we came across the term d value. So let's understand what is d value. So d value is basically the decimal reduction time, which means that time when the microorganism they destroy to 90%, and it is also the the number of the minutes required for the survival curve to traverse one log cycle. So if we take the semi-log curve and take the microbial population and time on the treatment time on the x-axis and the microbial population that's in the semi-log form. So the value this to this. So this is the on the x-axis, it's time. So d value is the time value when there is around 90% reduction in the microorganism from this to maybe this, or there will be when there will be 90% reduction, what is the time required to get it, or it is the reciprocal of the slope of the survivor curve and is measure of the death rate of a microorganism. Because why it's slow? Because this value, if we say, it will be this like this y2 minus y1 divided by x2 minus x1 slope. This is actually this value when the microorganism, this value when this will be minus to this, how much time this x2 minus x1 or t2 minus t1, what time we'll get is the d value. So d value equation is d equals to 2.303 by k log x by x naught. When the final population will be 90% reduced, that means 0.1% of the population will be there. So x naught is the total population and 0.1 is because it's the 10% only microorganisms survive in the final condition, like that the final stage. So final microorganism population will be x equals to 0.1 x naught. So if we put in this place x value, x equals to 0.1 upon 0.1 x naught upon x naught, so x naught will cancel, log 0.1 will be there. So after resolving, we will get 2.303 K. So we need to calculate here. Here we calculated till K value, right? But we need D value. So D value will be equals to 2.303 divided by K. So K is, here we calculated 12.28. So when we divide, we will get 0.1875 minutes or 11.25 seconds. Are you solving with me? Are you getting the same answer? So option A is correct. Is the solution clear?
Yes, ma'am.
Okay, so can we move to next one if the solution is clear?
We can move, ma'am.
Yes, ma'am.
So next question is
Ma'am,
about the
numerical questions are tough.
numericals, but you need to do, right? No option. So what you can do is you can practice more, right? Since we are discussing here the solution, the detailed solution, so you can understand one type of problem from here and you can solve from the textbook other problems, and it will help you, I think. So what is the generation time if 100 E. coli cells growing logarithmically for 6 hours produced 1.7 into 10 to the power 6?
Wait a second. Just wait a second. My Bluetooth is disconnected.
Hello, am I audible? Because I reconnected the Bluetooth.
Yes,
Yes,
ma'am, you're
ma'am.
audible.
Yes,
Yes,
ma'am.
ma'am.
Okay,
Yes,
thank
ma'am.
you. So we were discussing this question. Generation time we need to calculate here when we have E. coli as our microorganism and it is growing logarithmically for 6 hours and producing 1.6 into 10 to the power 6 cells. So we need to calculate its generation time. So the initial population is given as, which we are saying here, we can say N naught, X naught, anything. So initial population is given as 100 E. coli are initially present and it is growing. It is not here declining. It is growing and it became like 1.7 into 10 to the power 6 cells. From 100 cells, it grew and it became like so much cells, 10 to the power 6, and it took around 6 hours, and we need to calculate here the generation time. So the generation time, like what
Ma'am, not audible.
Oh God, why is this happening?
Ma'am, you are not audible.
Hello,
Yes, ma'am.
am I audible?
Yes,
Now
ma'am.
Yes, ma'am.
Now you are audible. Yes, ma'am. You are audible.
Okay,
yes, ma'am.
let me start recording again. I am so sorry from my side. Today, I don't know what is happening to internet. No, recording. Start. So, mm, share. So, yeah, can I start again from where we left?
Yes, ma'am.
Yes, ma'am. Yeah.
Okay. This slide
The
was
last question
we were
from the
discussing,
beginning
right?
of the equation, ma'am.
This slide we were right.
No, ma'am. Before the starting, beginning of the equation. This one.
This we finished.
No, ma'am. The next one.
From where the recording was like from where the teams was not
Yes,
connecting.
this one is over.
This problem okay, right?
Yes, ma'am. This we have finished it.
So, ma'am, you can start it from the generation time from the internet.
Okay, okay. So in the question, the generation time for E. coli is we need to calculate, where the initial population is given as n naught equals to 100, and the E. coli from initial population n naught 100, it grow logarithmically and it become like 1.7 into 10 to the power 6 cells. So it, and the time it took is around 6 hours, and here we need to calculate the generation time in minutes. So let's solve this question. So what happens uh, in the microorganism, since the organism it's a parent cell and it undergoes the binary fission and it is multiplying as two, like first one is converted into the two new daughter cells by the help of the binary fission, and then two will convert into another, each two means four, like this is multiplying, right? So for calculating the generation time, we need to calculate by this formula. G, which is generation time, is equal to growing time divided by number of generation. So growing time is, which is given as 6 hours in the question, that it is taking 6 hours to grow, but we don't know the n is number of generations. So number of generation is calculated by the help of this formula where NT is a final population is equals to the initial population into 2 to the power n. 2 because it is multiplying, and here n is the number of the generation, because it is multiplying with 2, then 4, then each the 4 cells will divide again into 2. So like this it is uh, changing, it is increasing its population. Here we don't know in this formula the n, so we are calculating value of n. So number of generations n is equal to 1 upon ln2 into ln n by n naught. So ln2 we have taken because one cell is dividing into 2 by the help of binary fission, and into n, which is the initial population, final population, which is 1.7 into 10 to the power 6, divided by the initial population, which was 100. So 1 upon ln into n, 1.7 into 10 to the power 6 by 100. ln2 is value is 0.693 into this, we can write as 17,000, ln 17,000, and when we calculate we get the number of generations as 14.05. So we now know how many generations it is taking to grow from 100 to 1.7 into 10 to the power 6 by when it is undergoing the binary fission. So it took around 14.05. This value is 14.05. Now the generation time is equal to the growing time divided by the number of generations. So the time it is taking is 6 hours, whereas it takes uh, for producing like it produces one generation, sorry, 14.05 generations it is producing, like the generation it produces. Mm, so G is equal to 6 divided by 14.05. So uh, generation time becomes 0.427 hours per generation, but we need to calculate in minutes, so we can convert into the minutes, which is G equals to 25.62 minutes per generation. We can multiply by 60 and we can convert hours into minutes. So is this question clear?
Yes, ma'am.
Yes, ma'am.
Okay,
Yes, ma'am.
thank you. Next, we are moving to the another numerical
Ma'am,
problem.
can we go to the backslide? I have a query. So
Yes.
does this formula holds for any microorganism, maybe yeast in fermentation, or any targeted microorganism in control condition in a bioreactor? So does this formula holds for every microorganism, or it differs?
Like when we are taking something like the microorganism, if it's growing by the help of binary fission, we need to see that which microorganism you are taking, and if it is going through the binary fission and doubling, so you can use this formula how it in like any condition bioreactor also you can take, and you can understand how, like from the initial population, the growth is there.
Okay, thank you, ma'am.
Yes, but it must undergo the binary fission.
So if any microorganism, this formula will work for the binary organ.
Can you please repeat?
No, ma'am. Is this formula help for the binary fission microorganism? It undergoes for that microorganism only.
Your voice is having some disturbance, so I'm not able to understand what you are speaking.
Ma'am, is this formula for the microorganism those undergoes only binary fission?
Binary fission. Mm.
Okay.
Okay, but most of the microorganisms they undergo binary fission. Uh, until they are not like they mostly they go like this. Oh, yeah, most of the microorganisms they grow like logarithmically they grow. Mhm. So next question is about again the the z value, and it is taking d value into the account in an experiment where the D121 value and the z value of a thermophilic spores in the whole milk was found to be 30 seconds. The D at 121, when we are applying 121 temperature, degree Celsius temperature, the D value of a thermophilic spore in the milk, it is 30 seconds, and the Z value, it is 10.5 degree Celsius. We need to determine D value at the temperature 150 degree Celsius, and the value should be in minutes for this spores. So first, we have already understood what is D value, but we have not understood what is Z value. So first, let's understand what is Z value. So Z value is thermal resistant constant, which is the slope of the thermal death time curve, where it is the Z value is the temperature, which can be in degree Celsius or Fahrenheit, and it is required to bring, it means when there is the in the semi-log curve of the TDT curve, when there is TDT curve means here it's D value on the y-axis, and on the z-axis it's, oh sorry, on the x-axis it's temperature value. So in the thermal death time curve, when there is reduction in the microbial population 10 folds or 90% reduction will be there, what temperature it it will take. So that is the Z value. In the decimal reduction, like 10-fold reduction in the decimal reduction time, the D value, in the D value time, when there is this the 10-fold change, when there, this is the D value. So when there is a 10-fold change in the D value, so at which temperature it is happening, which we will take it as Z value. So it is like this D2 minus D1, here T2 divided by T2 minus T1, like this, something like this, right? So temperature increase for a 1 log cycle change in D value. It means Z value is temperature increase for a 1 log cycle change in the D value. 1 log cycle or 10-fold change, anything. So Z is T2 minus T1, the temperature here T2 minus T1 divided by log D1 minus D2, because it's a, this axis is a logarithmic axis. So this is our Z value. But now in the question we have D value as 30 seconds at 121 degree Celsius. 121 means this is the autoclave temperature. So mostly we take T 121 degree Celsius is as reference temperature. So we write where, like as a subscript we write ref. So we are writing D temperature reference, which means D at 121 degree Celsius is 30 seconds. Temperature T ref is reference is 121 degree Celsius. T at 1, we need to calculate at T equals to 150 degree Celsius we need to calculate. So second temperature is 150 degree Celsius, and Z value is given as 10 to 10.5 degree Celsius. Now we need to calculate the D value at 150 degrees Celsius. So the effect of the temperature is given as this formula, right? Log Tt by D reference equals to minus T temperature at which we are calculating minus temperature of the reference, which is 121 degrees Celsius, divided by Z. From this equation, this formula, we rearranged and we are getting this formula. So from the rearrangement, we get dt equals to D reference 10 to the power minus T at 150 minus T 1 at 121 degree divided by Z value. So dt equals to, which is D at 150 equals to D reference, which is D at 121, which is given as 30 seconds. So we will put as 30 into 10 to the power minus T, the temperature which is 150 minus reference which is 121 divide by Z value, which is given in the question as 10 to 10.5 degree Celsius. So here 10.5 degree Celsius. So when we solve, we get 0.0519 second as our temperature, sorry, as our D value. D value is the time value, decimal reduction time, and it is in seconds, so we can convert it into minutes. So 8.65 into 10 to the power minus 4 minutes. So when we, it means at this D value means when we are heating at 150 degrees Celsius, so it takes around this much of the time to decrease the population. Before we were doing at 121 degree Celsius. So we were taking 30 seconds, but when we are doing at 150 temperature, the we are treating the thermophilic spores, we takes around 8.65 into 10 to the power 4 minus 4 minutes. Is this question clear?
Yes, ma'am.
Okay.
Yes, ma'am.
So we are moving to our next question now. A food item undergoes heating at 121 degrees Celsius for 25 minutes. To ensure proper cooking, we are treating this temperature and time combination. And this food, when we are cooking, it is going the degradation of vitamin C, which follows first order kinetics. So it is also following first order kinetics, the degradation of the nutrition, which is vitamin C. With a D value of 121 value, which is at 121 temperature, D value is 200 minutes. So we need to calculate percentage loss of vitamin C is there when we are cooking this food. So let's see. D value is given as 200 minutes at 121 degrees Celsius. So we can convert it into second by multiplying by 60, and then temperature, the treatment temperature, sorry, the treatment time is also given. We are treating this food for 25 minutes. So it means 25 into 60, so 1500 seconds, and the temperature is given as 121 degrees Celsius. And we need to calculate the percentage loss of vitamin C. So first we will see how much vitamin C is retained. Then we will calculate by minus from 100. We can calculate first, if we calculate how much is retained. So 100 minus retained,
Ma'am, can you please reverse the slide?
this one.
Yes, ma'am.
Can I move now?
Yes, ma'am.
So 100 minus retained is equals to how much lost, right? So first we will calculate the retained amount. So percentage, the amount of the vitamin C retained is equals to the final divided by initial equals to, since it is following first order reaction, so we can write in the same form like n equals to n naught e to the power minus kt, because it is the first order reaction. So we are taking here in place of n, we are denoting C, because we are trying to calculate the concentration of vitamin C. So the final concentration of vitamin C divided by the initial concentration is equals to e to the power minus kt. Is the reaction rate constant, and T is the time which we are using. So if we calculate in the percentage, we can multiply both 100, both side 100. So C by C naught into 100 is equals to e to the power minus kt into 100. The degradation rate constant, which is the k value from this, like the degradation, because in this equation we we need to put the k value here, this k value, but we don't know the k value. But if we calculate from the k value at 121 degree Celsius, we can calculate as k equals to, usually what happens, k is equals to 2.303 by d. It is the formula, right? So k equals to 2.303, and at the same temperature 1 divided by t, at the same temperature 121. So 2.303, and d value at 121 is given as 200 into 60, 200 into 60 seconds. So we can calculate the value of k as 1.92 into 10 to the power minus 4 seconds. Since we know the value of k, we can put now in this equation. So percentage of vitamin C retained is equals to e to the power minus k at 121 into t into 100. So E we calculate as, sorry, K we calculate as 1.9 92 into 10 to the power minus 4, and T, which is the time of the treatment, is given as we have treated for 25 minutes. So 25 into 60 as seconds we will put. So by solving this, we can get the percentage of vitamin C retained is equals to 74.98%. So, but we need to calculate the how much is the loss. So 100 minus 74.98, which is 25.02% as the vitamin C is lost during the process when we are heating, and when we are considering that this reaction or the rate of the loss is as a first order kinetics. Is it okay?
Ma'am,
Yes.
I have a practical question here. I'm not sure if this is relevant, but please help me out. So let's say
Mm.
I'm designing a product beverage. It's a orange juice beverage, and
Mm-hmm.
it again it goes through a pasteurization process. And if we use this theoretical formula to calculate, so just wanted to understand in practical situations when we are designing a product, Of course, we will be sending the final product for analytical testing. How closely this calculated results matches with real life testing?
I have not done any practical, like not any experiment, but what is my prediction is it will relate somewhat. But if there is any variation will be there, that is because of several other factors which comes in when you are doing as a in the industry. When you are doing it, then obviously the theoretical it's not will gonna exactly be the same because of the interaction of other factors also. But I am also not sure how much percent. I cannot tell you exact percentage that it is how much how it so like it how much percent it can be relate. But yeah, there will be obviously some variation, but not so high, I believe so, because uh, uh, I think the rate, because I think if there will be in the practical situation, I don't think it exactly follows the first order kinetics, because when we do the heating, uh, it can undergo second order kinetics as well. But it is
Okay,
we are
ma'am.
assuming this that it is first order kinetics.
Okay, okay. And ma'am, when we mention that in some product, when we see the label, it mentions that XYZ vitamins has been added to to compensate the loss during processing. So this is only
Right.
because of this, that's why we add this in
Right.
the label.
Right, right. Also,
Okay.
sometimes we add like orange is having vitamin C. Let's so right, it's having, but it may not have some minerals or some vitamins. And if we want to make some kind of energy drink, or which we want to feed the sports person or the person who are undergoing extensive work, so please can you mute? Please can you mute?
Question.
I'm sorry, please can you mute? Yeah. So
Ma'am, I have a question.
actually, I let me finish the explanation of the previous question, then you can ask. So yeah, when we are giving it to someone who needs extensive workout like that, so we intentionally sometimes like we say that it is having so many things, like it can uh, meet your RDA requirements, and it can have several minerals fortified, can have several vitamins as well. But yeah, sometimes we add intentionally just to uh, fulfill or just to compensate what we did in what we lost in the processing. So I think yes.
Thank you, ma'am. This was very helpful.
Okay, thank you. Next, your question, please. You may ask now.
Yeah, ma'am. Actually, it is a question of week one classes. That the
Yes.
percentage distribution of amylose and amylopectin in cereal starches.
Mm,
So it was given like it is 75% amylose and 25% amylopectin. Is it correct?
yes. 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 Mostly, but it also depends on like it depends on which cereal you are using, right? It doesn't and any of the food component, even its varieties, even if you are take if you even you take rice variety, different varieties, like you can take Indian variety, in Assamese Assam variety, some having this, in the Japanese they have this more sticky rice, so they have the, so there is a variation or a change in the amylose and amylopectin content even in the variety. So it's not like for all. there will be slightly change, plus minus will be 10 to 15% variation will always be there, even if you go
So
with the same
the
product with the
amylopectin would be lesser than the amylose.
amylose will be higher.
Yeah, no, actually, I googled out because it shows कि
Hmm.
amylose is 75% and the amylopectin is 25%. But in the
Hmm.
during the classes, it was given कि 75% amylose and 25% amylopectin. So in general, if question,
Amylopectin is lesser, right? What I know is amylopectin is in lesser amount and amylose is in higher amount. What I know is.
okay, ma'am.
So what I will do is I will reconfirm if there is any variation. I'll put on the discussion forum. But as far as I know, amylose is in the like higher amount and amylopectin is in the lesser amount. Is there anyone who would like to help us in this? Anyone, please help us. But I know this only. So if there is any variation, I will put on the discussion forum. Okay, is it okay?
Okay, ma'am, fine. Yeah, because
So
it's a
we.
Yes, ma'am. Yeah.
it's a fill in the blanks question. Like if something comes,
Hmm.
what
Yeah,
would
yeah,
be
correct.
75% or 25%? So it's a okay, ma'am. You be
Yeah,
confirmed.
yeah, I'll confirm.
Okay, ma'am.
Okay.
Thank
So we are moving to the
you.
next. I'll write somewhere.
Another question.
Yeah.
Uh, it's about
Yes.
myoglobin. Whether
Hmm.
it comes under storage protein or it's a transport protein.
Myoglobin.
Yeah, ma'am.
Hmm. I think it's because it's having the structure. It goes a pink color also. Muscle tissue. So I think it's a storage. I think it's my point of view. I need to check. I'll confirm you. Myoglobin, right?
Yeah, ma'am.
I think it's a storage, but I'm not confirmly like I'm saying. So
Yeah,
I'll
ma'am.
check this.
It is in between that only because on Google it shows ki like hemoglobin and myoglobin are the transport proteins. But
Okay.
when considering about the another aspect of myoglobin, where
Hmm.
it acts as an oxygen storage unit.
Yes, 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 yes. That's why. Hmm.
Like if it's for a conformation type, what kind of protein we can place it? Like it's a
Hmm.
storage protein or it's a transport protein?
Yeah, yeah, yeah. So I'll confirm your doubt. Give me some time because I want to check without. I don't want to
Yeah.
like just say anything and then which can be wrong. So I want to reconfirm before speaking anything.
Okay, ma'am.
Hmm. So we are moving to the next question. Calculate the processing time in minutes at 100 degrees Celsius and 150 degrees Celsius respectively. That would provide the same level of lethality as achieved at 121 degrees Celsius. This is based on the given values where F121 is equals to 4.5 minutes and Z value is 10.5 degree Celsius. So first let's understand what is F value. So F value is thermal death time. So it is also a time as D, but it is not same as D. So what happens here? Total time required to accomplish a stated reduction. So we say that from this to this we are decreasing, from n not to n we are decreasing our microorganism population. So how much time it takes? It is not like in the D value we say that 90% reduction is like fixed, but here it's not fixed, but it is stated in a population of vegetative cells or spores. So this time can be expressed as multiples of d value. So as long as the survivor curve follows a first order model. If a survivor curve is following a first order model, then f value is the multiplication of d value. Like for an example, we are saying 99.99% reduction in a microbial population. So it means that 4 log cycle it will reduce, or it is f equals to 4d multiplication of d, because it is taking the, like, it is decreasing the d value like 4 times more. So if we are saying 99.99% reduction, so f equals to d. Usually when we sterilize the food, or we say that if a food is a shelf-stable food, we consider the value f, like in the commercial level, we say that f should be equals to 12d. So 12d fold reduction should be there in the d when we are talking about the Clostridium botulinum as our microorganism. And here the second term we came across is lethality. So what is that? So lethal rate is the proportion of the thermal death time at the temperature T to the thermal death time at a reference temperature. So here we can see this curve where lethal rate is on the y-axis and time is on the x-axis. And in this, the area of this curve, the lethal rate curve, is known as the lethality. This is lethality. How much, like, total, like when we are applying the time and temperature, we are applying some temperature for certain time, then the how much population is decreasing. So it is the time at the reference temperature. When we are saying that at this reference temperature, we are saying at this reference temperature, what is the reduction in the population when there is another temperature we are taking. So what time it takes with respect to the reference temperature. So now our question, back to the question. So f value here is given as 4.5 minutes and z value is given as 10.5 degrees Celsius. So we need to calculate f at 100 degree and 150 degree Celsius. And we need to calculate the f100 and f150 degrees at 150, right? So how we can calculate if it maintains the same level of lethality? So the lethality is same, means the microorganism which are dying is the same, and we need to, like, tell at 100 degree how much is the time we need, and at if we treat at 150 degree how much time we need to apply. So the lethal rate is equals to, just now we discussed that the lethal rate is equals to the FR, which is the reference, the time of the treatment, which is at reference, which is 121 degrees Celsius, divided by the time, sorry, the temperature T, is equals to 10 to the power T minus T reference, which is 121 degree, upon its Z value. So for calculating FR equals to FT into 10 to the power T minus T reference upon Z, and from this we can rearrange again, and FT will be FR by, which is reference, at reference temperature, divided by 10 to the power T minus T reference divided by Z. So this is the formula for calculating the time when we have the reference temperature and the Z value. So we can calculate the F value, what time it will require for at particular temperature to get the same level of lethality as the reference temperature. So when we take T as 100 degree, because we need to calculate F at 100 and 150, so T at 100 °C. So F at 100 degree is equals to F at reference, which is 121, divided by 10 to the power T minus T reference, which is divided by Z. So F at 100 degree is equals to F 121, which is given in the question as 4.5 minutes. So here 4.5 minutes divided by 10 to the power 100 minus 121 reference temperature divided by 10.5, which is the Z value here. So F100 is equals to 450 minutes. So when we take 100 °C, so to get the same level of lethality as 121 °C, we need to spend around 450 minutes. Second case is a 150 °C. So we will solve similarly. Instead of like here, F121 is given as 4.5. So 4.5 divided by 10 to the power T here is 150. We are calculating at 150 minus T reference, which is 121 divided by 10.5, which is Z value. So F150 is equals to 0.0078 minutes. Is it okay?
Yes, ma'am.
This much is okay?
Yes, ma'am.
Yes, ma'am.
Okay. So last class we were discussing about Q10 value. So Q10 value is actually helps in predicting the, like it gives how much a chemical reaction, if it is a chemical reaction, so how it is going on. So it tells about that. So what happens? It usually helps in the shelf life prediction, like when there is some kind of spoilage in the food. That means some kind of the reaction is going on in the food, and because of that, uh, like at different temperatures, that reaction can be at different um, rate. Like if at 5 °C, the rate of the reaction will be lesser. Instead, if we talk about 30 °C, the rate of the reaction will be much higher. So their rate constant will also change accordingly. So it tells, like it helps when we calculate the Q10 value. It helps us actually if a reaction is going on at 10 °C and at 30 °C. So it helps to predict the shelf life, how like how much that product can sustain. So it will help to help in this. Usually, like when we increase 10 degree temperature, how its rate will gonna change. It is it is mostly related to the food, inside the food, how the reaction is going on. Whereas we discussed about D value, Z value, F value, it discuss about how we are decreasing the population of the microorganism in the food. So the topics we covered today are browning reaction, the enzymatic, non-enzymatic browning, food proteins, structure, denaturation, and its functionality. Then we discussed about the some part of preservation, the microbial growth, how the different factors affect the microbial ecology, and then we discussed about the D value, Z value, F value, lethality, the microbial survival curve, the TDT curve. All these we discussed today, and in the next class we will be discussing about high pressure processing, HPP, and the membrane technology. So you can see my recorded video here on the YouTube, and same I will be uploading in week 2 folder this week's presentation, and you can also write your doubts to me. One question I have not answered in the discussion forum yet about HPP, so I'll try to answer that as soon as possible. So yes, next session is on the next Sunday, 4 to 6. Thank you so much for connecting. If you have doubts, you can ask, or if not, you can leave the meeting.
Thank you, ma'am.
Excuse me, ma'am.
Thank
Thank you,
you.
ma'am.
Yes.
Excuse me, ma'am.
Thank
Can you
you, ma'am.
please share
Mm,
me your YouTube ID?
I have shared link here, right? If you go in this link, you'll directly get it.
No, I haven't received, ma'am. Sorry.
This is you. Can you see my screen?
Yes, yes.
The first one is my YouTube channel link. The second one is the Google Drive link. Can you
Okay.
click the picture? Also, in the Google Drive link, I'll upload this presentation. From that slide, you can copy paste. Is
Okay,
it
okay,
okay?
okay.
And I will solve the two queries I got for myoglobin and amylose and amylopectin. Is there any other query which I need to help?
No, ma'am.
Okay, thank you so much for joining. See you next week.
Okay, ma'am. Thank you
Thank
so much.
Thank
Thank you,
you.
ma'am.
Thank you so much for connecting.
you, ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you, ma'am.
Thank you. Whether final exam is online and same pattern as per the assignment. More recorded transcript.